Hello, I'm Dr. Mark Darcy, and this is Science on the Edge. In this episode, I'll be talking about the adaptive immune system. I previously recorded a podcast about the immune system in a very general way, partly the innate immune system, partly the adaptive immune system, but I didn't really deep dive. In this episode, I'm going to be focusing on the adaptive or the acquired immune system, and I'll go into a little bit of detail. So let's get started. Okay, so start at the beginning, the adaptive immune system. So here it is, or at least here is part of the immune system. So what we have here is the origin of immune cells. So we start off with various stem cells. We have stem cells in our bone marrow. And these stem cells, they then produce all of our blood cells. So for example, myeloid stem cells in bone marrow produce the erythrocytes, which are red blood cell. The megakaryocytes involved in blood clotting, they basically give rise to the platelets. The monocytes, which give rise to the macrophages. Macrophages are like local soldiers around our body in different organs and tissues. They respond in a very non-specific way to any invading organism or toxin, mainly by eating it. And various granulocytes. Granulocytes are a number of different granulocyte cells involved in the innate immune system. Won't go into those just now. We also have the lymphoid stem cells, which arise from the bone marrow. The lymphoid stem cells, they basically give rise to the B cells and the T cells. There are different B cells, there are different T cells. So B cells, B lymphocytes, they, when activated, produce plasma cells which release antibodies into our blood. We also have the natural killer cells. The natural killer cells, they sort of hunt around our body for any damaged or infected cells and basically tell them to kill themselves. They induce something called apoptosis, basically, programmed cell death. Now, some of the lymphocytes leave the bone marrow to go to an organ that's about here in our chest, known as the thymus gland. There they mature into T lymphocytes, such as helper T cells and cytotoxic T cells, which I will talk about a little bit later. Okay, so the adaptive immune system, it's a specific response system to specific antigens. So every organism, every cell, every bacteria, every fungi, every virus have antigens on their surface. These are unique markers. So coronaviruses have their own specific antigens. So do influenza, so does tuberculosis, so do our cells. Specific antigens coat our surfaces, and they can be recognized. Each T or B cell makes receptors on its surface that recognize only one specific antigen. So within our body, we have millions of different B cells, millions of different T cells, and they make different receptors that will bind to different antigens, like a lock and a key, or like a jigsaw piece being pushed into a puzzle. So they're specific. They're very specific. When activated, they can basically make clones of themselves. So if you activate a specific B cell by its antibody, its receptor, I should say, binding to a particular antigen, it will make many clones of itself, all that make the same antigen, that is, the same receptor that is specific for the same antigen. B cells will then release these receptors as antibodies into the blood, where they can seek out antigens on the surface of an infection and destroy it, or mark it for destruction. So the acquired immune system is very diverse. Collectively, the T and B cells have receptors for millions of different possible threats, millions of different antigens. They also have memory. Some of the B and T cells are made to a specific antigen, retain memory of a past infection so that they can respond to it again in the future if they are reinfected. So when you get an infection the first time, you might get sick. When you get infected a second time, because of these memory cells, you basically get over the infection pretty quickly because the memory cells wake up and start dealing with the infection immediately. So we can sort of divide the adaptive immune system into the humoral and the cell-mediated immune system. The humoral immune system, the B cells, basically. They are the cells that make antibodies that are released into the blood to stick to various infections. B cells can mature into plasma cells. A normal B cell has receptors for antigens, but they're on the surface. When it matures into a plasma cell, many plasma cells, many clones, these receptors can be released as antibodies to find the infection anywhere in the body. Some of the B cells become memory cells. In terms of the cell-mediated part of the adaptive immune system, we have T cells, helper cells, and cytotoxic cells. So helper T cells and cytotoxic T cells. The helper T cells, they sort of help the B cells to function, and they also help the cytotoxic T cells to function. So your helper cells help to regulate the entire immune system. And all of these types of cells can make memory cells and active cells. By the way, the cytotoxic T cells, their purpose is to find any infected cell and to destroy it in a slightly different way than the natural killer cells do. Okay, so to summarize that, humoral immunity is also called antibody-mediated immunity. With assistance from helper T cells, B cells will differentiate into plasma B cells that can produce antibodies against a specific antigen. The humoral immune system deals with antigens from pathogens that are freely circulating or outside the infected cells. Cell-mediated immunity is directed primarily against microbes that survive in your white cells or in normal tissue cells, normal cells of your tissues and organs. So basically, cell-mediated immunity is looking for infected tissues, infected cells, and it will destroy those infected cells. Cell-mediated immunity is also involved in tissue rejection in organ transplants, because the transplanted tissue will not contain the same antigens as the tissues of the recipient. Therefore, it will be foreign and there will be an immune response against it, unless the patient takes immunosuppressive drugs. So B and T lymphocytes, both made in the bone marrow, but the T cells are then processed in the thymus gland to make the helper T cells and the cytotoxic T cells. B cells produce antibody to antigen. By the way, the B just stands for bone. And they will basically release the antibody into the body, the antibodies into the bloodstream to find and stick to any antigen on any microbes and mark it for destruction. Cytotoxic T cells attack the invader directly. Helper T cells are involved in both processes. The cells are stored in the lymphatic system until required. The lymphatic system is a network of tubes within our body, which I will not go into in this particular talk. Okay, so now let's dive a little deeper. Adaptive immune system. So for those of you watching this on YouTube, there is a summary image here that breaks down the different components of the adaptive immune system. For those of you listening to this on a podcast app, I will be describing these systems in detail in future slides. Okay, so first step, recognizing the invader. B and T cells have thousands of receptors on the surface for one specific antigen. So each individual B or T cell has many receptors on its surface, but they're specific for a specific antigen. They bind in different ways, very different shape, basically, that matches a different antigen. Antibodies are these kind of Y-shaped proteins. Antibodies and receptors, same sort of thing, really. I'm using the terms interchangeably here. They're all sort of Y-shaped, but they have a different binding region at the end. Each particular antibody can bind to a couple of different antigens. Same antigen, but can bind to two of them and cross-link the four viruses, bacteria, etc., into a kind of gluey mess, which can then be marked for destruction. So how do B cells recognize an antigen? First of all, recognition of the antigen. The antigen and antibody specifically bind to each other, then different populations of B cells are formed for specific antigens. They then proliferate. How do T cells recognize antigen? Slightly different. So receptors on T cells recognize a fragment of an antigen. Antigen is then presented on the surface of an antigen presenting cell. So basically what's happening is you get a cell. The cell takes in the microbe, the virus, the bacteria, whatever it is. The virus or microbe will break down, and parts of that microbe, the antigens, will be displayed on the surface of the cell. So the cell is now an antigen presenting cell. So the cell has taken in the microbe, it is broken it down. Antigen is then presented on the surface of the cell, displayed in a complex known as the major histocompatibility complex. And then a passing T cell that has the right receptor, will bind to the antigen that is displayed by the antigen presenting cell and therefore become activated. So in a bit more detail, antigen presenting cells. So an antigen presenting cell might be something like a macrophage, for example. So a macrophage, one of those soldiers that guards different tissues of your body from infection. When you get an infection, the macrophage will eat that particular microbe, will digest it, will take some of the antigens from the surface of the microbe and display them on its surface in the major histocompatibility complex. A passing helper T cell with the right receptor will then come along, bind its receptor to that specific antigen that is being presented to it. This will activate the helper T cell. That activated helper T cell will now regulate the rest of the immune system. So T cells require antigen presenting cells to detect an antigen and to process it before they can recognize it. Then dendritic cells and macrophages are antigen presenting cells. They engulf antigen, degrade it into fragments. The antigen fragments are bound to major histocompatibility complex displayed on the surface. T cells bind to them, activate, and then release lots of chemical messengers into the blood known as cytokines that stimulate the division of both T and B cells. So helper T cells are absolutely essential for regulating the immune system. Okay, so once you've activated these T and B cells, you need to make clones of them, lots of copies of them, so you can deal with the infection. So first of all, you have a primary response. Some antigen is presented, you activate some B or T cells. So let's go with B cells here. You activate a B cell. When the B cell is activated, it will divide, make lots of copies of itself. Some of those cells will become plasma cells, which release antibody into the blood to deal with the infection. Other B cells will become memory cells and sit around the body waiting for the infection to come back. Once the infection is gone, if it does return, maybe years or decades later, the memory cells will wake up and there'll be a rapid response. Memory cells will make new plasma cells and release the antibody so quickly that you may not even get any symptoms the second time around. So selection and activation of a few stored B and T cells with the correct receptor is the first step. They stimulate, and they're then stimulated to proliferate, make clones of themselves that then will release the correct antibody. Memory cells will remain for the infection to come back. Okay, so let's break this down a little bit more, the B and the T lymphocytes. So you get some sort of antigen presenting cell, let's say a macrophage. It's eaten some microbe. It's displaying the antigens on its surface in the major histocompatibility complex. Helper T cell comes along, binds its receptor to the antigen on the surface of that macrophage, becomes activated. When it becomes activated, you release lots of cytokines into the blood that stimulate both T and B cells. The activated helper T cell will clone itself, make many copies of itself that will go around the body and activate B cells and cytotoxic T cells. The B cells will basically make antibody and release it into the blood, and the cytotoxic T cells will look for infected cells and destroy them. Okay, so let's first talk a little bit about B cells. So a B cell is triggered when it encounters its matching antigen. So you've got the receptor on the surface of a B cell. It binds to antigen from some infection. The B cell then engulfs the antigen and digests it. Then it displays the antigen fragments bound to its MHC molecules on its surface. When a T cell comes along, so a combination of antigen and MHC attracts the help of a mature matching T cell. This has to be a matching T cell with a receptor that will fit that antigen that the B cell is holding on to. This will stimulate the T cell to release cytokines into the blood to activate both B and T cells. But it will also stimulate the B cell in question to mature, to turn into plasma cells and memory cells and release antibody into the blood where it can find and destroy the infection. Okay, so once the MHC, major histocompatibility complex, and the antigen bound together, displayed on B cells, is recognized by a helper T cell. It binds together, it activates, releases its cytokines. So it's all about the B cell with its antigen binding to a complementary T cell with a receptor for the same antigen. This stimulates both the B cell to become a plasma cell and release antibodies, and the T cell to become activated, release cytokines, and activate both B cells and cytotoxic T cells. Cell-mediated response for T cells, how's that working? So this is against pathogens that hide in the cells. So the antigen-presenting cells, such as a dendritic cell or a macrophage, will present to a helper T cell, activate the helper T cell, which then, as I've said previously, will activate B cells and cause them to make plasma cells to release antibodies, but will also activate cytotoxic T cells. These cytotoxic T cells will then look for infected cells. They find an infected cell, a cell with the relevant antigen. They will stimulate that cell to die. They will kill it. So cytotoxic T cells. Cytotoxic T cell, when activated, will go and look for an infected cell. It will bind to it, if that cell has got the correct antigen, of course. And it will release a number of proteins which are toxic to the infected cell, causing the death of that cell. Okay, so I've sort of overviewed the T and B cells and how they work, but we must make a note of the fact that the immune system has memory. This is how vaccines work. So in the initial exposure, you're exposed to some sort of infection. You have a relatively slow response because you've got to find the right B cells, the right T cells with the right receptors. They've got to be stimulated to make the right antibodies, etc. So initially, response to a disease is fairly slow. But when you get that disease a second time, because you now have all these memory cells that were made from clones of the B and T cells with the right receptor, these memory cells sit around the body, and if you get infected a second time, they wake up, your immune system gets activated, and you deal with the infection before you even get a symptom, maybe. So vaccines basically, they use that process. A vaccine contains antigens for some particular infection, such as a bacteria or virus. Not the whole virus, a dead version of the virus, let's say, or a dead version of the bacteria, but the antigens of that. That will stimulate your immune system to make the right antibodies to become active, to make the right memory cells. And if you then are infected with the real version of the disease, then due to that vaccination, your memory cells are there, they will wake up, they will make the right antibodies, etc., and you'll deal with the infection very quickly. Okay, so to summarize, the adaptive immune system, it consists of both B cells and T cells. We have the B cells, which when activated will mature, become plasma cells, release antibodies into the blood that are specific for a particular infection. They will then create memory cells, which remain in the body until the infection comes back again and are activated very quickly the second time. Meanwhile, we have T cells, helper T cells, cytotoxic T cells. Helper T cells help to activate both the B cells and the cytotoxic T cells. They regulate the immune system. The cytotoxic T cells look for infected cells and destroy them. I should mention that these helper T cells, they are what are destroyed by a disease known as HIV. So HIV can destroy your helper T cells, and with those destroyed, you end up with an immune system that cannot be regulated and therefore become extremely vulnerable to any type of infection. So to briefly summarize, the adaptive immune system consists of T and B lymphocytes. It is specific, it regulates other components of the immune response, and it has memory. Okay, so that's pretty much all for now. The immune system is actually way more complex than I have just outlined. You can take entire degrees and PhDs in immunology. It's a complex topic, but this has been a sort of brief introduction to the adaptive or the acquired immune system. If you want to know more about me, then you can follow me on Twitter. My handle is @Macrophages, that's @MARKOFTHE or you can visit my website, scienceontheedge.com. If you'd like to help support this podcast, then you can do so by going to scienceontheedge.com and clicking on the donate button. Okay, so that's all for now. Thank you very much for listening. I look forward to speaking with you all again very soon. Goodbye for now.